Welcome back to another episode of Carterra's Scientist to Scientist series. My name is Alyssa Hughes. I'm an application scientist with Carterra. And today on the show, I'd like to welcome guest scientist Mike Brown. Mike is a senior scientist at Adimab, and he'll be answering questions today on high throughput binding characterization assays and antibody discovery. Mike, welcome.
Thanks for having me, Alyssa.
Yeah, absolutely. So let's get started. Kind of diving in, could you explain a little bit about what you guys do and the focuses that you have in the applications that you're running?
We're a protein analytics group at Adimab, and we've got about a little under 20 people who are split into three groups. We have an antigen quality control team, we have a mass spectrometry team, and those folks are really charged with quality control on our antigens, on our outputs from our selections, antibodies, antibody fragments. And then there's a sprinkling in of this developability characterization that has been big for the last 10 years or so. The mass spectrometrists work on chemical liability assessments, and then we all sort of pitch in on some of these developability characterizations like the thermal stability, the hydrophobic interaction chromatography, the AC-SINS assays. And then the group that I'm primarily associated with is the group that does most of the kinetics and affinity measurements and epitope binding. The application, at least on the Carterra side of things with the LSA that we use the most is the kinetics application, but we also do a fair amount of epitope binding as well. So we use both applications there.
With similar applications and comparable data, what advantage do you see in using the LSA over other instruments in your workflow?
Yeah, so there are a bunch of advantages to using the LSA. Your website does a great job of letting people know that you can run a lot of samples with very little antigen, especially on the kinetic side of things. So with the epitope binding, you do consume a little more antigen than on the kinetic side of things. But that also translates to savings on the other consumable side of things, the sensor chips, the sensor tips, that sort of thing. And so we see a lot of cost savings from putting things over onto the Carterra. The other of the more with less is that just running the experiments themselves are quite less labor intensive than some of our other platforms. And so you kind of have that set it and forget it type of experiment where in some cases, for a 96 by 96 epitope binding experiment, that experiment goes on for 3 days in some cases, and so a person can go on vacation, and so that's how set it and forget it the Carterra LSA can be at the extreme. And then the other thing is on the more side of things, it's by far our highest throughput instrument in the lab, and so we're able to collect more interactions per cycle. This 384 interactions in a single cycle is something it would take 4 instruments from one of our other platforms to do this, collect the same amount of data in the same similar amount of time. And so those are some of the main advantages I think that we get from moving things over to the LSA.
That's an incredible amount of work that you guys are able to kind of push through really quickly.
We're doing like 15,000 interactions a month on the kinetic side of things.
Oh, wow.
And we're doing a couple of epitope binding experiments. Some of these, sometimes they're 96 by 96, a month. And so, yeah, huge. We're cranking out a lot of data.
How does having the LSA in your characterization strategy kind of change the process upstream and downstream the way you guys work?
Sure. Yeah, this slide addresses it quite well. So one of the main advantages and on the upstream side of things is to be able to benchmark our samples much earlier in the process. And so Here's an example. I show on the left-hand side this idea that the Bucor and the Carterra can give you, basically the same kinetic parameters. And so for us, when we do collect Carterra data, we trust the data, and it allows us to, in the case of the sample, shown here, where we've got a benchmark control that's in the 200 picomolar range, and we see that, from whatever output, we're only at 17 nanomolar in terms of affinity, and we want to get to that benchmark. We know, and we can communicate that to our partner that, yeah, we're not there yet, but we know in a cycle or two we'll be there, and we'll have antibodies for you. So on the communication side of things, it's great, because it's key to, at least to our business, to be able to communicate results, intentions, as early as possible, so everybody, all other plans can be put in place for these samples later on. Again, on that upstream side of things, the ability to be able to test as many samples as we want, and test as many antigens as we want, and not really compromise at all on that side of things, is something new, at least using the Carterra LSA. A lot of times with our older platform and other platforms, we often have to make decisions based on maybe screening a single antigen, and then we have to cull samples from that, and then we're regridding samples in order to then test against some of these other reagents. And so in the case of using the Carterra LSA, we're able to just screen them all. And so again, set it, run it, go on vacation, whatever. The ability to just collect as much data as possible, and then also because we can make better decisions that way. And then the epitope, being able to provide a detailed epitope binding profile is more of a downstream thing. The more detail we have for a particular clone, the more information we have about how specific it binds, can help us make the best decision for us and for our partner.
Okay. In your talk at our recent symposia, you talked a little bit about your FC formatted antigens and how you're using those to improve connotation of weak binders. Can you talk to that a little bit?
Sure. Yeah. So for years, we've run this assay on our other platforms, and it takes, sometimes a couple of rounds to get, high affinity clones that bind to the antigen. And so for us, using this AVID format of the antigen allows us to pick out weak binders and out of the grass. And so here's this assay format is it's pretty straightforward. We are doing a capture kinetics assay on the Carterra. We'll immobilize the GOAT anti-human FC IgG on the sensor surface. And the idea is to capture the IgG of interest, and then block with an irrelevant IgG. And that should block all sites on the sensor surface, such that when you do come in with that FC fused antigen, even if it has the same FC, there shouldn't be any interaction with it on the sensor surface. And you can see down in the bottom left-hand corner, the ADDI IgG1, you see no binding, and that's a good indication that there's no binding of the FC of the antigen to the sensor surface, and that way you can trust the rest of the data and the rest of your output. And here we're able to rapidly tell whether or not one clone is better than another. We're not really worried about the intrinsic binding affinity in these experiments. We're really just relatively ranking things. And you can clearly see in this case, like ADDI IgG2 is a better binder than ADDI IgG3. And so we can communicate that to the partner, and they can make decisions going forward.
Yeah, that's amazing. I feel like being able to communicate those things early and like very directly is very helpful for sure. Could you share a little bit more about your experience in identifying more bins on the LSA versus other platforms, and why is that a benefit to your workflow? Why is that helpful for you guys?
Yeah, so the thing that gets people most excited about the Carterra and the LSA and the instrument itself are these heat maps. Partners get excited about them. Our campaign managers get excited because they just provide such a rich amount of detail on the binding properties of our candidates, and so I think it's probably the clear benefit is just having that much more detail. And sometimes these fine details can turn out to be so unique that maybe there's the opportunity to file for some intellectual property, that sort of thing. And so the more information we have, the better decisions we can make. And these heat maps often provide really enlighten us on what our output is providing us.
Thank you so much for joining us again today, helping us produce this video and sharing your Carterra story. We're excited to have you as one of our users.
Thanks. Yeah, this was super fun. Thanks for having me.
I'm Alyssa Hughes, application scientist at Carterra, and I'd like to welcome you to another Carterra Scientist to Scientist video. Our guest scientist today is Randy West Hansen, PhD. Randy is a senior scientist for antibody technology at Symphogen, and she'll be answering questions today on characterizing antibody libraries using HT-SPR. Welcome, Randy.
Thank you.
Let's get started. So first, thank you so much for giving such a great presentation at our recent symposia. It was really engaging. Could you describe a little bit on what your lab focuses on and the applications that you're running?
Yes. So as you just said, then I'm working as a scientist here at Symphogen in our antibody technology team. So at Symphogen, we are focusing on developing therapeutic antibodies, mainly within oncology and inflammation. So in our team, we are generating the antibody repertoires and then perform high throughput screening to characterize the antibody and then down to the final lead. So this is basically showing our workflow in our early discovery process. So we get our antibodies from B cell, from immune donors, and then isolating the B cells, and then we generate recombinant antibody libraries by sorting the B cells, and then making the antibodies by our simplex technology. So here you can see that we start around 10,000 recombinant antibodies. And then when we are about down to around 400 antibodies, we start to characterize the antibodies by SPR, where we use the Carterra LSA and has been working with it since late 2019.
What is the average timeline through that process with the LSA? I mean, what's the average time you're spending on HT-SPR going from that 400 to 1?
I think it really depends on what kind of assays that we perform. So on the next slide, there's an overview of the different assays that we do when we characterize the antibodies by SPR. So we do, in the beginning, when we are around the 400 antibodies, we do the affinity ranking and also, of course, the binding kinetics of the antibodies, and also do cross-reactivity to orthodox antigens. And I would say that these basic affinity assays, in principle, we can perform it within a day or two. But if you need to have some optimization and also the analysis itself, I would say that you need at least a week, maybe 2 weeks, also because often we have other tasks to do. But the focus time on the LSA, that can be done in a week.
Wow, yeah, that's incredible.
Yeah.
So can you describe a bit about some of the epitope work that you do and mutant mapping data and things like that, and how that impacts your decision making for antibody selection?
Yes, so we both do epitope binning, which is this competition assay where you get an idea of how the libraries, how they cover the epitopes of the antigen. And then later on in the process, when we have identified the final lead, we have this epitope mapping where we take the antigen and then mutate all the positions of the amino acids, so do an alanine scan, and then measure again where the antibody, where they start to lose the affinity for the antigen, and thereby characterizing the epitope of the antigen. So I think in the epitope binning, there we will get like the broad overview. of how our antibody covers the antigen, which we then use in the selection if we want to target a unique epitope and also differentiate for if there is any competitors. And then for the epitope mapping of the final lead, we mainly use that for IP. So that is to have a stronger patent when you can characterize the epitope as well.
Okay, yeah, that's yeah, your work is incredible. It's very cool to get kind of down in the details of that. You mentioned a little bit more of your cross-reactivity work. Could you kind of dive into that a little bit and describe how you screen for cross-reactivity on the LSA?
Yes, so we do it as a first pass affinity, we call it. So it is when we are here at 400 antibodies. So this example here shown on the slide, I think it's around 200 antibodies. But when we're setting up the affinity assays, we set it up so in a full assay that we first measure, capture the antibodies and then measure the affinity to the human antigen, and then regenerate the assay and capture antibodies again from the same plate, and then measure affinity to, for example, the cyno antigen, and then again to the mouse. So we in one assay can get both the affinity and also the cross-reactivity to for example, these 3 antigens shown here on this slide. So here we can use it for our selections if we want to have cross-reactivity to, for example, cyno and mouse. And then we can already early on in our projects, we can start to design the in vivo assays later on. And also if we have any tox, or we always have tox studies, but also so we can prepare for the tox studies later on.
Okay. So last slide that we've got to cover with you, can you dive into a little bit more on your high throughput epitope binning? Specifically, if you're losing 10 to 20% of your antibodies, or they're becoming inactive during immobilization, how does that affect your data analysis?
Of course, then we will not have the epitope information on those antibody. But again, I also think here that it can be project specific how many antibodies we actually lose, because we often do this high throughput epitope binning, as we call it, quite early in the projects as well. So it's not always that we have deselected the antibodies with loose binding. So this can also be part of it. But I still think that we're losing 10 to 20% upon immobilization, then we will not have the data of these antibodies and get this idea of the epitope coverage. But again, we have a lot of other data, so we have both sequence data and functional data and binding data. So still sometimes also from the sequence data and clustering of our sequence data, we will have an idea of where we think our antibody binds. So so often we do not use that much time on optimizing the conditions early in our projects. Yeah.
Yeah, and it seems like that really kind of improves the quality of your analysis too, kind of narrowing things down. Could you kind of describe some of the key attributes that you uncover using the LSA to identify therapeutically relevant antibodies?
I think that the key contributes from the LSA is that we quite early in our screening processes can get the kinetic binding information of our antibodies. And also this epitope binning, I think that's also have a very high value to our projects, and especially in this also work with bispecific antibodies, where you really want to cover and test different epitopes on the antigens and then see how they can work together. Then here it's have a really high value that you can get some of this information early on in the projects.
I want to thank you so much for participating in our Scientist to Scientist series. I know you're super busy. We appreciate your time and input in helping us develop more content that can assist others in understanding high throughput SBR and how it's working in real labs around the world. Again, thank you so much for your time. We really appreciate it.